Hi all. Today we are going to see about system suitability parameters in HPLC. We are going to understand one of the most important concepts in HPLC analysis. System suitability parameters. That is called system suitability parameters. Now don't worry about the name. It may sound technical, but in reality it is very simple. Before we start any HPLC analysis, we must make sure that our system is working properly. Just like before driving a vehicle, we check the brakes, isn't it? We check the fuel and we also check the working condition of our engine before we are leaving using our vehicle. In the same way, we want to check the HPLC system using some parameters that is called system suitability. And how do we check it? By reading something very important, the chromatogram. So we will see the chromatograms shape, size, and we will see the quality of the chromatogram, and we can find any fault in the HPLC or not. Okay. So in this session, I will show you how to read chromatograms and understand whether your HPLC system is ready or not. By the end of this video, you will be able to look at a peak like this, which is shown here, and immediately understand what it is telling you. So let's start step by step. First, we need to understand why we do this check. So as a first step, the laboratory pre-flight check. So before we start analyzing any sample, the first question we should ask is, our HPLC system ready to give correct results or not? This is where system suitability testing comes into the picture. You can think of it as a health check for the HPLC machine. Just like we don't directly start a journey without checking our vehicle, in the same way, we should not start analysis without checking the system. So, system suitability ensures that the instrument is working properly before we run actual samples, especially important when dealing with pharmaceutical or patient samples. It also gives confidence that the results we obtain are reliable both scientifically and legally. Think of it like a pilot before takeoff. They always check everything to ensure safety. Similarly, in the lab, this is our pre-pilot or pre-flight check. Now the question is, how do we actually check this? For that, we need to understand the chromatogram. Okay. So, to check the system suitability, we need to clearly understand the chromatogram. So to understand the chromatogram, we want to see this slide reading the map chromatogram basics. Think of a chromatogram like a map that gives us complete information about what is happening inside the HPLC system. Now let's break this map into two simple parts. The x-axis represents time. It tells us when a compound comes out of the column, so it helps us identify what the compound is. The y-axis represents the detector signal. It tells us how much of the compound is present. So remember this simple idea. X-axis tells us what and y-axis tells us how much. Every peak is giving us both identity and quantity at the same time. Now that we know how to read this map, let's look at our first parameter, that is called retention time. So retention time simply means the time taken by a compound to travel from injection to detection. In this chromatogram, the injection starts at 0 minutes and the peak appears at around 5 minutes. You can see it in the chromatogram. This is the retention time. In simple words, it is just the travel time inside the column This parameter is very important because it helps us identify the compound. If a standard drug always appears at a specific time, then a peak at the same time in the sample likely represents the same compound. Think of it like your daily commute. Reaching at the same time confirms you followed the same route. So retention time confirms identity. Now that we know that compound is the next question. How much of it is present? For what? Sorry, for that we look at peak area. If retention time tells us identity, peak area tells us quantity. Peak area is the total space under the peak curve. A larger area means more concentration of the compound in the sample. You can think of it like a bucket of water. A bigger bucket holds more water, just like a bigger peak area represents more drug. So, while retention time answers what is it, peak area answers how much is it. Now we know identity and quantity, but what if two compounds come very close to each other? Can we still measure them clearly? That brings us to resolution. Now we want to see about the resolution. Resolution tells us how well two nearby peaks are separated from each other. If the peaks are too close and overlap, we cannot measure them accurately. This is poor resolution. But if there is a clear gap between them, we call it good resolution. The goal is to achieve baseline separation where both peaks are separated completely in a distinct form. Think of it like personal space. If two people stand too close, they blur together. If they stand apart, you can clearly see both. Similarly, in chromatography, we want clear separation. Now, even if peaks are separated, we also want them to have a proper shape. That brings us to the next parameter called tailing factor. So what is the tailing factor means? Tailing factor measures how symmetrical a peak is. Ideally, we want a perfect symmetrical peak, but sometimes the peak stretches more on one side. This is called tailing. This usually happens when the compound interacts too strongly or gets slightly stuck inside the column. Think of it like a runner dragging a parachute. The front moves fast, but the back lags behind. That creates an uneven shape as shown in our chromatogram. In HPLC, we want clean, symmetrical peaks because they give more accurate results. Now, along with the shape, we also want sharpness. That leads us to our next parameter, theoretical plates. What is that theoretical plate means? It represents the efficiency of the column. In simple terms, they tell us how sharp and narrow the peaks are. A higher number of plates means sharper, taller and narrower peaks, which is ideal. A lower number means broad and less efficient peaks. Think of it like cutting bread. A sharp knife gives a clean, thin slice, while a blunt knife gives a thick, messy slice. Similarly, a good column produces sharp peaks. So now we have understood all the key parameters. Let's quickly summarize everything together. In this system suitability scorecard, you can see all the parameters as a summary. So here is a quick summary of all the parameters. Retention time checks identity. We want stable timing. Peak area checks quantity. We want accurate measurement. Resolution checks the separation. Tailing factor checks the symmetry. And theoretical plates check the sharpness. Okay. So if all these parameters meet the required criteria, we can trust the data and proceed with the analysis. But if even one parameter fails, we must stop and fix the system before continuing. This is very important in pharmaceutical analysis. Now let's see what a perfect chromatogram looks like when everything is correct. You can see a perfect chromatogram shows everything working correctly. Stable retention times, accurate peak areas, clear separation, sharp peaks and perfect symmetry. When you see this kind of chromatogram, it means your system is healthy and your results can be trusted. So now you know how to read a chromatogram and judge whether your system is suitable or not. But what happens if things go wrong? What if peaks are broad, taily, or overlapping? In the next video, we will learn about common HPLC problems and how to troubleshoot them step by step. Okay? Thank you, all of you.